very much, Walter, for the generous introduction. Can everybody hear me in the back? Okay, good. Thank you. So yes, just like Walter said, this is an interdisciplinary group project that included several members, and I would like to acknowledge everybody on this slide and thank everybody who contributed to everything that we are going to discuss today. So in addition to myself, Chuanzhu and Feng, there were several core members who are faculty at Wayne State University. They're listed here alphabetically. So we have Hayung, maybe you can wave, just Jeff, Liliana, Martha, and Peter. Feng was very important. He did all the statistical analysis with all of the data that compiled. We also have several students that worked on the project. Some of them already graduated and moved on to Ph.D. programs. Some are still up with us. And so you will see Shahmardan, Yamei, Jinhan, and Mardeya helped out. And we have a couple of external members who have been consulting on the project as well. And we would like to thank the Humanities Center for their generous support. We actually didn't realize we could apply for another year of funding. We thought it was limited to just the 2. So that's why we didn't apply. The plan for the talk, I would like to, or we would like to cover these 3 questions. So I will start by asking, or talking about the question whether there are meaningful individual differences related to language ability among humans, because this is actually a kind of a controversial issue, and I will frame it in terms of, I will frame it in a way that I've learned from Karen Stromswald many years ago, that she came and gave a talk at the University of Connecticut when I was a student, and she's one of the few linguists that are interested in the genetics of language. And she, when she was presenting, she asked the question, is language like fingers, or is language like height? Meaning that if language is like fingers, it's pretty much equal in all human beings, because the way the human hand works and looks is pretty much uniform, with only very minor differences. Or is language like height, so we are all different, and there is large individual variation. So I will talk about that, and there are 2 positions. Some people try to defend the idea that it is like the hand, others point out that really there are differences in language capacity among individuals. And then the second question that I would talk about is whether there is a possible relationship between any of the differences that we can identify in language capacity and the genetic basis that underlie those differences between individuals and differences among populations, so from an individual level to the population level. And then finally, whether comparing populations that speak typologically different languages with an idea that we can learn something about genetics of language that possibly led to development of these differences, typological differences in different languages. And so as part of that, we will discuss the results of the study that we've done, where we attempted to do just that. Compare languages that speak typologically different languages, look for the genetic differences between them, and see if we can link differences in the language features with differences in the genetic makeup of these populations. So, is language like fingers or like height? So, like I mentioned before, there are 2 views. The universalist view, language is universal, and the capacity for language is uniform among humans. At least the way it's presented, they always use the word in the relevant sense. In the relevant sense, in the important and the fundamental sense, it's the same. And the differences, of course, there are differences, but they're superficial and unimportant. They don't go deep. The opposing view is the variationist view, that there are differences and they're important, and they go back to the genetic differences. So differences that are neurobiological, differences in terms of brain functioning, and those are due to genetic differences. So the universalist view would see language like fingers, that if you're born normal, then you would have your 5 fingers and your hand would look like this, with very minor variations. And only in some rare cases, a single genetic mutation will cause your hand to have an extra finger. It's called polydactyly. So it's rare. It's categorically different and very easy to see the distinction. It's either normal or abnormal, and it can be traced to one single genetic mutation. So they would like to think that language ability is invariant, at least qualitatively in the relevant sense, in all normal humans. So language disorders would differ from language normality in a categorical sense, like the 2 hands. And so it's always clear-cut. You can always tell this is normal, this is a disorder, and they would like to trace that to clear single genetic mutations. Because those genetic mutations will show you the genes that are involved in language. And a celebrated case of that was a big breakthrough in the early 2000s or the late 90s, the KE family that a lot of popular press wrote about. It was a 3-generational family in the United Kingdom. Many members, approximately half of the members in that family, had a severe language and speech disorder. And you could tell just by looking at the pedigree how this was transmitted across generations that it was genetically based. And so this family was studied extensively, behaviorally, neuroimaging, genetics, and they in fact identified a single mutation in the gene FOXP2, which is the most famous language gene. A single mutation caused this severe disorder that was transmitted with this mutated gene across 3 generations of this family. So that universalists rejoiced and announced that yes, languages like this, and we found the first language gene, and then maybe we will find maybe a couple more or a few more, and we will know all of the genes that are involved in language. It didn't really pan out like that. So, of course, this is an important gene, but in most cases of individuals with language disorders, you cannot trace it to, there is nothing wrong, there's no mutation in the FOXP2 gene. So most run-of-the-mill cases of developmental language disorders are not due to this mutation. This is in fact rare. But in fact, people who study language ability in humans from this different perspective, from the variationist perspective, would like to say it is more like height, in that there is a normal individual variation, and so you can rank people by height. So this is all your favorite male Hollywood celebrities ranked by height, and so from the tallest to the shortest. So it's a measurable trait. Language is measurable. It's not all or nothing. It's on a continuum of ability. So variation is in fact continuous. There is no sharp distinction between tall and short, but it's a full continuum. Also, like height is highly heritable. We know that our height is similar to our parents, so we inherit our height. Heritability estimates are upward of 0.8. That means that 80% of variation in height is attributed to the genes. It's rare mutations that cause extreme short and extreme tall stature do exist. There are disorders like that, dwarfism, but they're very rare, and they certainly do not account for these kind of variation. And this is kind of an interesting thing. So they've discovered about 50 genetic variants associated with normal variation in height. They all contribute to the differences in height, but altogether they only account for about 5% of phenotypic variance, meaning the difference in height. So why is that? It's called missing heritability. It's a big puzzle in genetics. So if we already found 50 genes that contribute to differences in height, but altogether they explain only 5% of that difference. And it's not really clear, and maybe the biologists can explain it better, but perhaps 50 is just a very small number. Maybe a lot more genes are involved and contribute to height. An effect of which is so small that each individual gene contributes only a negligible effect. So we can't really appreciate the effect of all of the genes because we haven't discovered them all, and that's for height. And plus genes interact with other genes, and genes interact with the environment, and those interactions are hard to detect. So something like this must be true for language as well. Language, so the brain is a very complex organ. The way it develops and functions is largely attributed to the genes, and 75% of all genes that we have in our genome are expressed in the brain. So hundreds of them are involved in how our brains are built during development, and many of them, or maybe most of them, have something to do with how we function in terms of language. And so we only scratched the surface because I will show you we didn't come up with nearly as many as 50 variants of candidate genes for language. It's maybe more like 10 or 12. So that's the variationist view. So language is like height. Perhaps language, maybe it's a false dichotomy. There are certainly universals that we can talk about, but it's also true that there are differences. And in order to discover genes that underlie language capacity, we have to look at differences because it's impossible to, so that's basically a requirement. People have to differ in the phenotype, in the traits that you are studying, and to find the genes that cause that difference. So basically it's impossible to find the genes that create language universals because they're all the same. The genes are the same and the phenotype is the same. So that's the avenue to look for genes, is to look at differences. So language acquisition is variable, it's measurable. The whole field of speech pathology is based on the idea that you can measure language consistently, reliably, more or less objectively. So it's measurable, continuously varying. It's heritable. The heritability estimates vary between 50 and 75%, so about half to 75% of the variance in language ability if you measure it in the ways that it's measured using tests. For disorder phenotypes, so if you characterize people as affected versus unaffected, the difference, 50 to 75% of that would be attributed to genetics. For normative sample, it's less, between 34 and 39. But basically, the idea is that language capacity varies. It follows this normal distribution, like all other traits, like height. Most people fall in the middle, so the middle of the curve, it's the tallest. That means most people fall here, in the middle and right around the middle. So it's 68% of the population, and then 95.5% fall right here. There are very few on this extreme ends of the distribution. So these rare extreme cases that perhaps are caused by mutations like FOXP2. In most people, including those who would be called affected, who fall here, who would be diagnosed by speech pathologists, they would be here. And there is still relatively many cases like that, and they're not attributed to any rare mutations in single genes. So that's just, maybe I'll go through this quickly. This is data from about vocabulary size in very young children. So this is their age from 16 months to 30 months. And they're divided into quantiles. So the green line, these are kids who perform in the 50th percentile, so they're average kids. And then these two lines are the two kids who are in the 75th and 90th percentile, so above average. And then these are the two bottom, 10th and 25th percentile. So you can see how they're differentiated. Starting from 18 months, you can already see the differentiation. How much variability in how many words a toddler knows. At 20 months, some of them go, some dots are right here, hundreds of words, and others are here, basically a handful or to no words. That's how much variability there is. And none of these kids would have single mutations. You would not find anything genetically wrong with most of them. So now, what was discovered in terms of language genes? I took this from a recent review paper. The title is here. It's too small. It was published in 2016. The evolutionary history of genes involved in spoken and written language beyond FOXP2. So what else? What other genes are involved? So these are the names of the genes. You don't really need to know just to see how many. It's not that many. It's a lot less than 50. This is the name of each gene is involved in, has instructions for building a specific protein. And so if there is a mutation, there is an error, the protein doesn't work out. And then there is a disorder phenotype. LI stands for language impairment. DD stands for developmental dyslexia. So compromised ability would be, they call it language. So oral language, vocabulary size, syntactic complexity, however it was measured. Frequently it's just measured by people repeating long nonsense words. That's a pretty good marker. It differentiates people pretty well, even though it's a very simple phenotype. And some of these language genes were discovered by looking at reading capacity, not even spoken language, but actually a lot more research has been done with reading. For some reason that I'm trying to understand, I don't fully grasp why reading has been more productive in terms of search for genes than oral language. I would like to maybe add this gene that I was fortunate enough, I was in a team. We worked on a genome-wide association study looking for genes for language, and we've discovered, so we published a paper where we've discovered this gene, SEDBP1. We studied this isolated population that resides in Russia in a far north, far away from other dwellings of humans, and because they're isolated, it was slightly easier to look for this gene among them. And they have a high frequency of language disorder among children and adults. And so we published a paper where we reported this gene. And so the disorder would be LI, language impairment, and the phenotype that we used to link with this gene was syntactic complexity, basically counting how many complex sentences they use in a narrative. All right, so next we can ask, perhaps it's true that there are meaningful differences in language ability, but could we also say that there are meaningful differences among populations? Can these differences be clustered within populations? So height is continuously varying, but if we measure height in people from different countries, and if we look at the average height, we could see that, so this is according to Google, the tallest women would be in Norway, Russia, and Italy. And there's a continuum of, so next is Spain, Germany, and Greece, etc. And then India is on the bottom. So can we maybe see if individual differences can be clustered within populations in a way like that? And of course, there are tall and short women in all of these populations, but the average comes out like this. So next I was going to talk about, are there meaningful differences among languages first? So if we simply look at languages, Steven Pinker in his famous book, The Language Instinct, said, according to Chomsky, a visiting Martian scientist would surely conclude that aside from their mutually unintelligible vocabulary, Earthlings speak the same language. And people who work in that tradition are convinced and they're excited about the fact that they think that all languages are the same. And the universality is what's interesting about them. On the other hand, there are, on the opposite end, there are people like Evans and Levinson who said that, of course, this widespread assumption that languages are the same is completely false. The true picture is very different. Languages differ so fundamentally from one another at every level of description that it's very hard to find any single structural property they share. So the opposite view, they have nothing in common. Which, of course, it seems like a rational view to say, yes, languages have universal features. They are also very different on many levels, and it's interesting, perhaps, to look at both. So, some of the differences. There are large differences in the realm of phonology. For example, just how many consonants a language has. Some languages, like this one, spoken in Papua New Guinea, has only these 6 consonants. On the other hand, this African language from Botswana has 122 consonants. They have a large number of different clicks. So a child that has to acquire phonology here versus here has a very different task, level of complexity. Of course, all children can learn any language, but it's not all children really, because approximately 10% of the children in every population are children who have a language or speech disorder. So perhaps this it would be interesting to see if the complexity of the inventory changes that number. Some languages also has unusual consonants, like uvulas, pharyngeals, front-rounded vowel, etc. Differences in syllable structure from very simple CV open syllables to highly complex, with complex onsets and codas with multiple consonants, up to 3. In Russian it's going to be 4 in the onset. Languages have tone or don't have tone. Differences in stress, how complex, predictable the stress pattern is, if it's completely idiosyncratic or if it's completely predictable, if it's based on rhythm or not, if it's sensitive to weight or not. Morphology, also a realm where languages differ very widely from languages that are isolating, where all formatives are funnel, each formative is a word. There are no morphologically complex words. Versus concatenative languages, where grammatical formatives, morphemes, bound morphemes are attached, phonologically bound to a host. It's very simple to very complex paradigms. To languages like Semitic languages where morphemes are non-linear, so their formatives are not even realized in a linear sequence, and they are never encountered by children who learn them as autonomous phonetic entities. So in an Arabic word, there is a triconsonantal root, and morphemes are interleaved within the consonants. So the vowels are the grammatical, they call patterns. So they express some grammatical function. They're interleaved into the consonantal root so that it's modified to get you. So words like, so the root, these 3 letters with these vowel patterns will give you a word like maktub, which means written, and with this pattern it gives you the word for writer. And so these consonants, the root has the meaning, something to do with writing, and how you modify it changes based on what pattern you insert within the root. But the point is that children never, to learn what the root is, what the pattern is, and how that changes the word systematically. It's a lot more complicated than in a language that's isolating or has a simple, regular, concatenative pattern. Grammatical categories. A language like, so English doesn't really have any cases for nouns. There are some remnant case system for pronouns, but nouns, the noun is always the same no matter how it is used in a sentence. But so the number of cases ranges between 0 to 15 to over 15. So this is Hungarian. It has the most complex noun case paradigm of all languages, according to Walls. It has 21 productive cases. So this is a word for ship in Hungarian, and depending on how you use it in a sentence, it might have one of these 21 forms, and a child would have to master this system. There are also different noun genders from 0 to 20. Hungarian, I think, doesn't have genders, so luckily, if they also had 20 genders, then this would have to be multiplied by 20. But for example, Russian has 6 cases and 3 genders, and so each gender would have a separate paradigm for case. And the verbal domain, of course, is notorious for being so different across different languages. So in English, if we talk about present versus past tense events, we modify the verb. So students eat pizza versus yesterday the student ate pizza. In languages like Mandarin and Indonesian, there are no grammatical markers, so you would use the same verb in both sentences without any changes. In Russian, you would mark the change, and in the past tense, you would also have to indicate the gender of the subject. And also, there is an obligatory marker that you have to add to indicate whether they ate all of the pizza or only some of the pizza. Turkish marks evidentiality in some other languages. So if they say something like, oh, so the student ate pizza, if you saw it with your own eyes, if you eyewitnessed the event, it would be marked in one way. If it's through hearsay, it's marked a different way. In Arabic and Hebrew, there is polypersonal agreement. The verb doesn't just mark subject-verb agreement, but also object-verb agreement. So he sees him versus she sees him versus he sees her. The verb would always be marked differently, depending on the gender of the subject and the gender of the object. And there is also polysynthesis, like languages like Nahuatl, where all of the formatives and all of the arguments can be incorporated into the verb. And so you can express one complex sentence that in English you would have to say something like, I shall make somebody give something to you. It's all incorporated within the verb, and it's just one verb expressing this whole complex sentence. So obviously there are differences among languages. So people who are interested in the differences between languages frequently talk about this in terms of how language must influence thought. There is a relationship between complexity in language and complexity in cognition, and so this is a celebrated example of that. So language, this language spoken, it's an Australian Aboriginal language. They have no egocentric frame of reference, so they don't have words for left or right. When you describe location of an object, you can't say it's on the left or on the right of someone. You have to use geocentric frame of reference, to cardinal points. You always have to say north, south, east or west, even if you're talking about a very small, like if I were to say the bottle if I wanted to express the location of this bottle, I would have to, relative to my computer, I would have to know whether it's north of my computer or south of my computer, and of course I have no idea. But apparently, even 5-year-old children who speak that language can point to the north, anywhere, including in an enclosed room with no windows. So obviously there is something special about the speakers of that language, and the idea that people usually, when they talk about it, say, well, it just shows that they use this language with the terms and it trains their cognition and they become excellent at pointing out cardinal directions. But I would like to suggest that maybe it's actually the other way around. Somehow something genetically predisposes them for having excellent prowess for spatial orientation. And that's why their language came up with the system where they use, instead of left or right, they use north and south. So the direction can be not from language to thought, but from genes to brain to thought to language. And so this is our hypothesis that we took kind of as a starting point for our study, that language is shaped in part by the production and perception systems. And cognitive system, perhaps there is language-specific, domain-specific language system in our minds and our brains and underlined by our genes, but also working memory, procedural memory, etc., different types of memory. And then individual variation in these domains has a neurobiological basis, so it can be traced to brain anatomy, and that can be traced to genetic differences. So there are these genetic variants that are multiple. We don't really know what all of them are yet. They confer subtle biases for processing certain types of linguistic complexity at different levels, for phonology, for morphology, for semantics, for syntax. And how these genetic variants are distributed among world's populations is not equal. There are more speakers with these variants in some populations than in others. That's why there are taller people in some countries and shorter people in other countries. It's due to differences in genetics. So we would like to maybe use that to try to find language, try to find language genes by comparing people who speak languages that differ with respect to certain linguistic parameters. Some have the value plus and some have the value minus. So you can think of it, some people speak a language that has greater degree of complexity in one domain, and others speak a language that has lower level of complexity in the same domain. And maybe comparing them genetically, we can find where the genetic basis for these differences. So I don't know, maybe I should, if we are running short, I can skip the study. This is the study that was done inspired us, that was very similar. Do we need to start talking about our... So this paper by Didier and Ladd, they looked at distribution of linguistic tone on this map. This shows languages that speak tonal, populations that speak tonal languages, so the black squares here and here, and the yellow languages that have no tone. And then they looked and they found 2 genes and specific variants in these 2 genes. This is one of them, ASPM, and the other one was microcephalin. And they found that there are 2 alleles, so 2 variants in these genes. One is the older one, and one is a newer, mutated one. And how the new derived, what they call it a derived allele, is distributed lines pretty perfectly. So the concentration of that allele is higher in, so the darker circles show high concentration of that new allele. And the pale is very low concentration, and the low concentration aligns pretty well with tonal languages. So they showed that there seems to be this relationship between these 2 specific variants, the new derived alleles, and speaking a tonal language. So now we took that up and we started our study. We got together and we said, well, that sounds very interesting, so let's do what they did, but bigger and better. So we first brainstormed, and everybody in the group came up with between 1 to 3 different linguistic features. We then we characterized, we found a number of populations, and I'll explain in a minute how we characterized them with respect to this feature in a binary way. Each population was labeled as a plus or minus for evidentiality, whether they speak an unusual consonant or vowel, whether they have ergative case or accusative case, etc. Then we retrieved genetic information for these populations. We identified informative genetic markers that we know have something to do with brain and language. And then we try to find for a relationship between speaking a language with a plus value or a minus value in each feature and any genetic markers that we could find. And we attempted to control for shared geography and language family. So I don't know, there's probably not time. I'm going to skip the slides. I was going to give you like a basic information about genetics. How much time do we? So
it's 1:15. 1:11.
Okay, so yeah, okay, so maybe if you want to ask me, I can explain those 2 slides, or you can look on your own. So I just want to move on to the nitty-gritty of the study. So we use these 2 publicly available databases. One is called WALS, World Atlas of Language Structures, where this was our initial source for all language information. And Alfred is allele frequency database. Allele is a genetic variant, a gene, a piece of the DNA that can come in different forms. So I have to say that. So the DNA is a really large molecule. It's 3 billion base pairs. It's really long. Somebody estimated that if you print all of the DNA on paper, it would be something like 245,000 pages long. But there is only less than 1% of that sequence can be different from human to human. Most of it is the same from human to human. In fact, between humans and chimps and humans and broccoli, a lot of it is just always there. But so about 500 pages of that book would be only the pages in which people would differ. So we have to look at those 500 pages. And so the allele is the name for the part of the DNA that varies, that is not always the same. So Alfred is the database that's publicly available, and they publish information about frequency of different alleles in different populations. And so we took that, we took all the populations that they had, and then we just excluded those that did not correspond to a clear language, that maybe was a mix like North American English, includes many different people, it cannot be used, but more or less clear-cut correspondence between the population and a single language that's spoken in that population. And so we characterized them, and this is what we came up with. This is some of the features, and you can see how many different populations we characterized across all of these geographical regions: Africa, East Asia, North America, Oceania, Siberia, Europe, South America, and Asia. And then we did genetic analysis, and so I'm Sorry for taking so much time.
Thanks for Dr. Natalia's informative and interesting introduction. Now let me talk about the genetic analysis to determine the association between the genetic factor and the language characteristics. So for our genetic analysis, we mainly have 3 steps. The first step, we access and extract the SNPs because, as Dr. Natalia's introduction, for the R3 database, it have a few hundred populations, and each population have 1,000 of SNP informations. So that if you multiply it, it will be in a now fashion world, it's a big data. So we want to try find the available SNP data which can be found in both groups, plus or minus. And in that groups, they should have at least 2 populations have that SNPs information. So we first extract that SNP information, then conduct the test to find any SNPs can show the statistics, the significance or difference between that 2 groups. But for that SNPs, we want to find whether that SNPs is related with the language-related genes or not. If that is related, we call it informative SNPs. Then we want to exclude the effect of the geographic and linguistic relatedness. So we built regression models to exclude that effect. Try to assess the association between SNPs with the language characteristics. The SNP. The association of the SNPs and the language characteristics. So that's 2 main words, and let me use that illustrations. So beginning, our partners already categorize the groups. Then we exercise it, extract it by step 1, so we can find the SNPs which are available in both plus and minus. Then search for informative SNPs which show the statistics significant. Then we confirm the SNPs with our GE models.
Can I just say what a SNP is? Because I don't think I introduced the term SNP. So SNP stands for single nucleotide polymorphism. When you look for differences across the genome at the level of 1 single letter, it's a SNP.
Okay. So let's continue.
Because gene can have, it's long, it contains many, it could be hundreds of letters, but we're looking at individual letters.
So let's continue for the go to details about the statistics test. We extract all the SNPs which can be found in both groups, minus and plus. And for that SNPs, it should have 2 allele information. So like allele 1 is A, then allele 2 is C. So for that SNPs, it should have some numbers of A and some number of C. Then we calculate some that the population which in the group minus, how many A it have, how many C it have, and same as the plus 1, how many A have, how many C have. Because it's 2 tables, so it's contingent tables. We can use that Fisher exact test to calculate the p-value to say whether the association between the SNPs with the language characteristics is significant or not. So as this one showing that we find, I think for the parse and non-parse, we find 16 informative SNPs related with the language-related genes. And I will talk 3 which may have functions that Dr. Feng will talk later in our later slides. And for that 3 SNPs, the first one, RS2243381. So 4 populations show the minors of the non-parse and show that non-distinctions. And 5 populations show the distinctions. And for that one, so the first groups, it have 87A, it have 47G, and the other groups, it have 259A and 23G. So we can calculate the p-values, and for the p-values, I think the take-home message is if less than 0.05, then it means significant. But the disadvantage for the Fisher exact test, because it can produce some false positive result. Then we continue to do the Q-value test to get rid of the false positive result. The same as p-values, so q-value is minor, is smaller than 0.05, is significant. So both numbers show that the association between these names with the language characteristics is significant. They may have association. And same as the other two, so the both p-value and q-value is significant. They also have associations with the parse and non-parse distinction or not. Okay, now we know that three SNPs have associated with language characteristics, but we need to consider other facts whether impact the language characteristics. The most important thing is geographic and linguistics relatedness. So for that one, we first need to calculate the degree of geographic and linguistic relatedness between each pair of language in the analysis. Then we construct the matrix and construct the matrix, then put that coefficient data into our statistical models to exclude that factors, geographic and linguistic relatedness. Try to see whether the association is still significant between the genetic information and linguistics characteristics. So thanks, Martha, and we can build up this geographic distributions for that 9 populations, and you can see it's widely distributed. And the blue means it have distinctions for parse and non-parse, and right it means no distinctions between parse and non-parse. Then according to the geographic and linguistics relatedness, we can categorize into 4 groups. And they are 0, which means different language family, different geographic regions. 0.25, different language family, widely separated within same regions. 0.5, same language or same geographic region, but not both. And 1, same family, same geographic regions. So we build that matrix, then we use that generalized estimating equation. That one is to try to say whether the probability of the association between the language characteristics with the SNPs still exists, still significant to controlling the effects of the shared linguistic lineage and geographic. So you can say these equations, we put the correlations between populations. We consider that part. Then after this equation, we can calculate the coefficient of the 2 alleles, A and G. And we also can calculate the standard errors. Once we have that 2 numbers, we can do the Bohr test to produce the p-values. The whole take-home message is same as the previous test. P-values. If the p-value is smaller than the 0.05, that means the association between that 2 alleles is still significant related with the language characteristics. So all that 3, you can say from the p-values, excludes the geographic and language relatedness. The association still exists. So that can prove that our method is correct. Then I will leave Dr. Fan to talk about the function of that 3 SNPs.
Yeah, so I'm going to finish up just a couple slides left, so to talk a little about biology. So, wrong way. So just talking about, we identified the 3 SNPs. So those 3 SNPs are just by name, don't worry about names. So just RS, always use RS, we call it ref SNP, so and then by the numbers. And the 3 SNPs, we look in 2 genes. So first gene called DC, DC2, DC is a gene name to the acronym for the double coating domain containing 2. So this is just a gene name, and then another gene is a KIA. So these 2 genes, these SNPs are looking at 2 genes, and then also we're looking, because we're looking for informative SNPs, which means we're looking where the location of those SNPs. So in that particular case, in 2DC2 gene, this thing we're looking called a 3-parameter UTR region. So those region, the region we're looking for, because the gene is very long, so some region doesn't matter, some regions really matter. So we're looking the region which maybe matter for the genes, which potentially, if have mutation in that region, it can cause transcription or some like a defect for the genes. So this is the case because those two snippets looking the UTR region, so which means if a mutation happens in that region, it potentially can cause a defect of the genes. So the second one, I mean the third one also for the 1GKR gene, also looking for the splicing region, which means also can be a defect. So for the genes, and also both of those location, there's 2 genes in the chromosome 6, and also both genes is a gene something related to neurodevelopment. So usually causes this language-related genes, and then the gene can cause a potential reading defect or speech defect. So, and then always go one way. So, and then if you look at the function of the 2 genes, I mean the function which I said, both 2 genes is for neurodevelopment. So if something genes happen in the developmental stage, which may cause the phenotype for the particular people, like you have reading defect or speech defect. So which another thing is because both genes look in chromosome 6, so they are very close by. So they're very close, only very far away for 2 genes. So the one thing I just finished. So which means that's like we are only looking at like a non-population right now. So which looking for the past tense parameters and then for non-populations. And I was starting, I mean, this is working in the progress, and then we're starting to collecting more data in the case. So we had 47 populations. So this 47 population have the 3 SNPs. So we try to collecting whether they have all the data for the past or non-past in the case. So, but we still try to collect the data here. So I mean, the blue is already, we identified there, the blue is, there's a distinction, red and no distinction, but still the gray, which means still unknown. We try to working hard, so all the linguistics, Martha, Liana, and Hai, and Peter. So, and then we also try to getting like external help. We go to online to post and then try to identify, because I am not a linguist, I don't know exactly how it works, but actually, language is very complicated. So in the case, we try to identify all these features, and then this is the current stage, which means we have more data right now. So in terms of the 3 SNPs, and then we'll identify more than 10 to 15 populations, either in distinction or non-distinction. So, I mean, we just said, just working in progress, and we try to collect the data. We increase the population size, and then we can redo analysis, and then see whether still has can see this significant association between the 3 SNPs and with the past tense parameters. So, I think it's the last slide. We're talking in future directions. So. And I just mentioned we continue to collecting the missing linguistic information for past tense parameters, which I said will only identify something 20. So right now we have 47 populations still missing something 20 populations, so we still don't have the data. And then also we're just talking about, right, because we're looking for the genetic analysis, we're only looking for the candidate gene right now. So we're looking for something 20, 30 language-related genes. So we identify the SNPs. Actually, we are not just doing this like candidate genes. We are doing the genome-wide association study, which means looking whole genome, human entire genome. Natasha just mentioned about it, because in human there's 30 billion base pairs, so there's 20 to 25,000 genes. So, and then in that case, we potentially can identify not just those like 20, 30 language-related genes, or potentially can identify the other candidate genes. Those genes may be also linked with the language like parameters. And then last thing is we are just talking about past tense parameters for this like talk. And that's more than 20, I think it's more than 20 like language features. So we are just continuing to do the analysis for each individual features, and then we identify whether there's like a genetic factor associated with the particular language features. So which means there's just in progress. In the future, we will potentially getting some publication to combine all the data we have, the genetic data, all the language feature together, and try to get in some publication. This is our plan. So I think the last slides. So I finished up. I think for questions.
I have a question. Earlier, reference was made to nations and the mix and the comparison of the height differences were made. And I thought to myself, you know, people have been mixing for thousands of years that come from many different areas. I mean to say that Germans have this height and Italians have that height. I mean, they're nations are fictitious entities. They're not real things. It's that you can't compare them to like a race. You know, like we say race. And so I kind of wonder about them in my head. That's my first comment. But my other comment is that if we, I think of this in another context when we say that if there is a genetic thing, if that could be used like race against them, well, these people don't have enough linguistic skill because they have this linguistic deficiency and these people don't. So those two things I would make for my comment.
Okay, I'd like to maybe answer that. So the first question, nations, right? So that's why we don't really talk about nations, we talk about populations. And some are very heterogeneous, like the United States would not be a good place to study population genetics because it's so mixed and there are immigrants, but other populations are relatively homogeneous. And so, for example, well, Russia is a multinational, multi-ethnic country, but you could study, if you use the word population rather than nation, you could study entities that are smaller than the nation, ethnicities, something that's more homogeneous genetically.
Well, I guess I'm also thinking that it's like, is it like, since you talked about the brain, how do you not confuse that with brain differences? I mean, if we're saying that.
It's just a different level of description. It is. Okay, so maybe I should address the second question. Yes, it's very easy to kind of fall into the scientific racism kind of frame of mind and say, well, this population is superior and this is inferior. This is really not at all what we want to say. What we want to say is that certain genetic variants give a very subtle and small bias towards processing certain kind of information. For example, greater capacity for processing greater articulate sounds that are more difficult in terms of articulation, right? And so this bias is relatively small and relatively subtle, but over generations, perhaps it can give rise to these features that the language is more likely to use more complicated sounds if people are more predisposed to being able to use these sounds than a language where these genes do not, people don't have those genes and cannot easily produce the complex phonetic sounds. But this bias is so small and subtle that it still allows any child to learn any language on a kind of on a population level. So it's not an absolute, it's not a great distinction. It's a small, subtle distinction. And also, it's not that one population is superior in all respects. We don't want to say that. It's just that all languages are complex, but they are complex in different domains. They are not all complex in every. They are not equally complex in every domain. Some languages have higher complexity in phonology, but simpler other morphology, something like that. So it's not. We don't want to have this kind of statement that some populations are superior in terms of language than others.
I didn't say you would say that.
Or we don't want anybody to say that.
Somebody else, because we have just a little bit of time. Hold on, before. Somebody else who's not members of our family.
Yeah.
Questions?
Oh, there's over there.
Well,
Yeah.
No, you go first. I'm gonna
No, no, go ahead.
You go ahead first, because she works for the Humanities Center.
So I related to your question. I think that we don't necessarily have to talk about more complex versus less complex, but rather different in some aspects. And actually, evolution is very much about losing as it is about gaining certain features. So in fact, when we study tone, when tone was studied by Badiou and Ladd, it's actually tone is frequently lost in populations. So, so the idea is that if you lose some of the language features, then over generations you become less capable of making those distinctions. So in fact, there was a proposal that in language evolution there was a stage of a so-called musical language, where there was a little bit more melody involved, but that over the evolution this was lost, and that as a result we have all become less musical now. So now there are pockets of very talented singers, but very few of us are actually capable of that. So the way we see it is not that there is one kind of progression to the pinnacle of language complexity, but rather just little adjustments to the changes that occur, and they could be not just gains in a certain feature, but they can actually be losses in a certain feature. Just like over the language evolution, we have over the human evolution, we have lost certain abilities for smell, for example, etc., which maybe weren't that important to us anymore as they are to other species. So I think if we see it as a kind of little adjustments, little adaptations to the changes, to the language sort of niche in a way, then we really shouldn't see it as some kind of more complex versus simpler languages or features. So I think that's sort of where we are, the way we think about that.
Okay, go ahead.
Me? Okay. So I'm not really that great at like biology or chemistry or whatnot, but I kept on thinking about the liger, how you can make a liger, but the liger can't reproduce. Do you think that you can make like you say, not a race, but maybe like, a region of people who get all the languages and gets all the genes, but they lack somewhere else? Like super language, I don't know how to...
Super people.
No, you don't see you gain a lot of genes. It's just something we're talking about the alleles, like snakes, there was some mutations happen. So just single letter, right? DNA we call that A, T, G, C, we have 4 letters. And any of the letters have mutant called mutant. If change some other letter, potentially affecting the gene functions, maybe you are losing some ability, or sometimes the mutation maybe you gain some ability. So it really depends. So if you gain this ability, which means you are like a high intermediate evolution body called it fitness, which means because you're superior to other people, which means you can produce more offspring, which means you can increase like this kind of mutation into the population. Okay. So the usually what happen. Even though in the human evolution, because the human is not like we're not talking like animals, so the organism, because human has a social, so which means not pure natural selection. So human is a bit different because the social impact there. So and you're looking for the natural selection, which means it's pretty true. Some mutation will happen. This mutation is bad, which means that you're going to die, which means not going to produce any offspring, so you're going to go. If the mutation is good, so which means I am stronger, which means I can get more like as maize, I get more offspring, so you potentially you are increased mutation in the population. Eventually, you're getting all able death. I mean, back like the language feature, I think pretty much a similar thing. I guess you're thinking everything should be eventually contribute to genetic. So even though all the features are affecting the brain, I guess, right? So all the brains, which means change some genes, expression of the genes, which means you have ability to getting some other features. So if the particular group, they like that kind of people, so it's only the people like have the particular features, they want getting together, eventually, all the people have the particular like tones, whatever thing. So I want to get in there.
Could you go back to the slide where you sort of summed everything up and there was a whole lot of information on the right?
Which one?
That,
This one?
no, yeah,
Oh,
wait.
you're moving down. Who's, you do it. If you know which slide.
We're talking this.
Go later. No,
This
sorry,
last
go
one.
earlier. Okay, what?
This one.
What was on the slide?
Yeah, I guess that one.
This one.
Okay, so explain that a little more. You went too fast for me.
I mean, this is just we're just talking the function, the two genes, right? We're talking the two genes called the KIA and then DC. This was the function, was like a function in terms in the system. So I mean, usually I don't think we because you guys are not biology, don't worry too much. So that gene for the transmembrane adhesion molecule receptor guardian axon to appropriate the target, which means what I mean this like for neurobiology, so neurodevelopment. So which means they are going to respond for the neural, for the axon developing, axon elongation. So
So
if
can you
we
say
lack this,
anything that would connect that to the past, non-past thing? Or is that too?
It's too close.
Okay.
I mean, it's too far.
Okay,
yeah,
gotcha.
yeah. I mean, yeah. We just said those two genes you become called language related genes. So if something was to happen, those two genes, your potential can change your language. We don't say language
So
disorder.
you know what the genes do, and you know that they have a distinction for past, non-past, but you can't just tell me
We
what,
can,
why
I
that.
think we cannot say the cause.
Okay, gotcha. Okay.
Because you can never, so genes do specific things and the phenotypes are far removed from, you cannot really say this gene gives you past tense. There is a lot more
Okay.
distance between
Yeah.
that. But look, it's involved in axon growth. So axons are part of neurons, these long branches that branch out and connect with other cells. So that is really relevant, right, for functioning of the brain and acquisition of language or processing of language. Another one, what's another function? Axon growth.
Yeah, this
is
table
one.
for the axon growth. So DC also from the mortality of the developmental neurons. Pretty much same thing. This like take from the neurons.
Motility is how, what's motility?
Yeah, I mean, kind of just
Oh,
like
how
movement,
fast they
yeah,
move.
movement. So I mean, we
So-
just give the two genes. We don't see that the gene cause the past tense, okay? But
Right.
things are another
So
thing is that the
it's-
genes can have many color alleles.
Okay.
So some of those like this alleles may be very subtle phenotypes.
Okay.
So
So they make the brains function in ways that are slightly different,
Okay.
and somehow that over generations gives you past tense.
Okay.
Over there.
Yes.
So you looked at genes and sort of brain function, brain morphology. Did you consider the genetic impact or the impact that genes have on physiology, like physiology of the throat and mouth, that would cause you to be able to articulate or even hearing cause you to be able to perceive tone?
I guess we are not like a particular like in terms of a future study where there's like I mean we're starting with language related genes, see whether there's anything we can find for the language related gene. I mean actually eventually we are identifying for whole genome wide. We're going to identify a number of those genes and then we can characterize the genes, so see what the particular gene. We are not looking particular function like physiology or anatomy, but no, I don't think we go that direction. So we will do the whole genome screen. So, I mean, we are doing right now, so we identify the number of genes, and then we can categorize function of the genes, say some gene maybe. I mean, again, because this we call, those kind of trees called quantitative trees.
Right.
So, there's not single gene can respond for the trees. It should be like a very, I mean, talking height, right?
Sure.
Even those height is like only 5% for some gene. So, height is very quantitative trees. So, each gene only contributes less than 1% to the variability. So, and there's 100 genes in the height. I mean, language is pretty much the same thing because of quantum tree. So not just single gene should be 100 genes. Each gene only contribute very small, subtle. This
But I think maybe there is work like that, what you were asking. I think other people probably, it's probably more straightforward because it's a lot less complex than looking at language. Trying to link language capacity with genes is a lot harder because language is so abstract. People don't agree what it is or how to measure it, or if it's measurable. But something like musculoskeletal functioning is a lot more simple. I'm sure there is genetic work on that, and I'm sure genes have a lot to do with how, you know, how much athletic ability you have or how good you are at, you know.
Over there. You had a question?
Oh, yes.
I was kind of curious. I studied French briefly, and you know, well, I'm certain it happens in other languages that they put the noun, you know, the verb, or the noun before the verb, you know, well, whatever. And so I was just kind of curious, like in English, it's the exact opposite. You know, we put the adjective before the noun.
Mm-hmm.
But anyways, I was just wondering, is it easier for, has there been any studies on whether or not children, when they're learning language at, you know, in the earlier stages of their lives, do they learn it more easily if in the way English is constructed with the adjective before the noun, or does it matter, or if they put the adjective after the noun? Which one, in which language do children develop a language that, you know, acquire more words, and, you know, according to you know, the different stages of development?
So if you talk about word order, it's one of the things that children acquire very early, and equally well in seems to be in all languages. And in some languages, like English, it's fixed, so you would think it would be relatively easy to learn the word order. But in other languages, it's flexible. So there are many different word orders that they encounter, but children learn the word order very early in all languages. So it doesn't seem to make any difference.
So their language skill ability is about the same amount.
So there is that. You cannot deny that it's not. So we are not. Language is also like fingers. There is a lot of universality in how it develops in children across all cultures. It just, there is both. There is universality and there are differences. But I think a lot of it has not been studied. So because a lot of the languages that we are looking at are very, faraway languages that we don't know much about. A lot of acquisition data comes from English and a handful of European languages. They're all very similar to each other. Who knows what happens in these languages that, languages in Africa? Maybe it does take longer for a child to acquire phonology that has 122 consonants. It takes up to the age of 7 or 8 in English to be able to pronounce all the consonants. So we don't really, I don't think we know a lot. And it's assumed that it's equally easy to acquire any language, but most of the data comes from European languages.
I was just curious. Thank you.
Do you have time to go back to those slides you skipped over where you were going to talk about snips? Remember you said I'm going to skip over those?
Oh, it was just
explain
it took
something
a lot
that I don't
longer
know.
than I thought. I was just going to. It's just maybe it's not really relevant anymore because it was just some basic information about the DNA before they were going to go into the complex stuff.
Yeah. Yes.
We kind of. I passed it.
Yeah.
It's right here, so
You got to those 2 meaty slides and then passed on. Oh, not that.
it was just
This little ball.
No, not
it's
that
this
one.
very basic baby course
Yeah, not that one. Go to the next
Not
one.
that one. Well, the next one was to introduce
Yeah.
this concept of SNPs. So
Okay, so do snip for a minute.
I mean, do you want to? It's so the DNA, and then so each individual the part. So maybe I have to go to the previous one first.
Okay.
Because so the DNA is this long molecule that's packaged in chromosomes, and it's a double helix. So everybody knows that. So it has this structure. The backbone, kind of the skeleton, is the alternating sugar and phosphate groups, and each sugar has the bases attached. And then there are 4 bases, so the 4 letters. There is the A, C, G, and T. These are the 4 letters that the whole human DNA and all living things are written with. And so A always bonds with T, and C bonds with G, right? Is that correct? Okay. So, and this sequence is invariant so less than 1% of the sequences would be different. And so all of the differences between people come from less than 1% of the DNA. And when you look at the, an allele is this parts that come in alternative versions. And if it's at the level of a single nucleotide, so like a single letter, then it's called SNP. And a lot of the work in genetics, it doesn't look at genes, because genes are really long and large, but they look at variation at the level of single nucleotides, and that's what
Yeah,
they
so like
Those three things you had were SNPs, is that right?
Yes.
So I mean,
SNPs and two
Yeah.
genes.
so this gene usually contains something 1,000, we'll call it base pairs, 1,000 letters.
Right,
Majority of the letters is exactly identical between you and I.
right,
But you see some of the letters are different.
right.
So even though less 1% different. So those different we call SNP.
Right.
So for example, here we have 5 individuals for the particular genes. I mean, whatever the gene is. I mean, I just list like some 20 letters. All the letters are identical, but it's only one of them. So this is number 5. Number 5. So they're different. Like the first 3 have T, the other 2, 4, 5, C. So this T and C we call SNP, called single nucleotide polymorphism.
Okay.
And so how does it come about that there are two instead of just one? Probably in the course of the human evolution there was a mutation.
Yeah.
So one of them is usually called the ancestral and older one that maybe other species also have. But then there was a mutation in some individuals that, and if it was selected, positively selected for, if it gave you some advantage in one area, and then it spreads, and so now there are 2 variants, and some populations have a lot, so this new allele, say C is the new one, and some population, more people there have this new one compared to other populations.
So usually the SNP sometimes can determine a phenotype. So why we are able to look different. So majority of those different come from those SNP, even though you have some like deletion insertion also happen.
You had 3 SNPs that you showed us for
Yeah.
your past,
Yes,
non-past.
yes, you remember that, yes.
Okay.
So we identify 3 SNPs
Okay,
in the 2 genes.
okay.
Yeah, so
Yeah.
2 of them come from one gene and another
Yeah,
one from
yeah.
another gene.
Yeah.
And also I want to just add, so a mutation can give you a positive effect, but at the same time it could also give you a disadvantage. So for example, certain variants were linked to better working memory, but at the same time they're also linked to higher risk for anxiety disorder. So it's not always, if it's a positive, it doesn't mean it's just great in everything. It could also give you some disadvantage.
Isn't it also true you can have a variation that you don't know if it is expressed at all?
Mm-hmm. Yeah.
That's
So
true.
it's-
Some mutations have no effect at all.
Or you
Yeah,
don't-
actually, once most of this mutation
Yeah.
doesn't matter.
Okay.
We call it
Right.
neutral.
Yeah,
Mhm.
okay.
Only small subset of this mutation, maybe.
Yeah.
It's the real reason you have somebody like born as disabled because of something the gene was happened, so you are affecting the genes, so they're born as disabled, you know. There's some, you know, yeah.
This is so exciting what you guys have found.
Oh, thank you so much. Thank you for thinking it's
Thank you.
exciting.
Well, I guess, right?
Thank you.
That's their view as the piece.